welcome to Chapter 17, Selection and Evolution, Part 2a, Natural Selection. Now, in this particular chapter, we'll be learning a lot of things, okay? First of all, what is natural selection? What are different types? What is the importance of genetic variation in selection? And examples, as well as Hardy-Weinberg Principle, which is a little cute maths thing, which is very easy, don't worry. Then we're going to learn genetic drift and founder effect, and finally, artificial selection. So let's start with just the first part here, natural selection. So here's an introduction and a few terminologies that you need to know. Now, previously we've all been talking about individuals, like what phenotype does this rabbit have? What phenotype does this particular crocodile have? Or what phenotype does this particular human have? But now we're going to change our lenses, okay? Stop thinking in terms of individuals. Think in terms of population. A group of individuals of the same species living in the same area that can interbreed. Okay, so no longer thinking about individuals, thinking about how populations change together. Now, in this particular chapter, these words become very important to you. Number one is alleles, different forms of a gene. Allele frequency, next one, right? Allele frequency is a number of occurrences of an allele in the population. How frequently is the allele found in the population? You can see here that the allele frequency changes across the generation here. Okay, small a, not so many at first, and then it becomes more, and generation 20, everyone has the small a allele. So this allele frequency has increased over time. Now, this little circle here represents the gene pool, which is all the genes or alleles present in the population. Okay, and you can see how this gene pool also changes across a generation. How does it change? Well, the allele frequency in the gene pool has changed. So that's how you use those words. You will see these words pop up, and hopefully you'll get what it means in a deeper sense as we go through this particular video. Okay, now that you have learned those terminology, let's talk about these two fellas here, what they do, and what the heck is natural selection, right? What is it? So let's talk about this first. Theory of evolution. This is the idea that organisms change over time. Evolution, evolving, right? It literally means change. And not necessarily change species, but form varieties and species over time. And this forming of different varieties and species is actually due to change in alleles frequency over time, right? Many millions of years, many thousands of years. Now, one mechanism which evolution can occur is through natural selection. Okay, and this theory of evolution by natural selection, again, is a mechanism of how evolution occurs, is proposed by Charles Darwin and Alfred Russel Wallace. And these two fellows actually came up with it independently, and they actually wrote each other letters, and they went like, oh, we actually came up with the same conclusion by studying different things independently, which is this idea of natural selection. And Darwin, after 1858, they proposed a paper, okay. Darwin published the famous On the Origin of the Species the following year. So yeah, that's a little introduction to natural selection. But what is it still? Okay, we haven't really explored that. So let's talk about it. What is natural selection? So natural selection was actually deduced, okay, or they came to this conclusion of natural selection through some series of observations. So here are some observations. Observation number one is overproduction. Alfred Wallace, as well as Charles Darwin, looked at many different organisms and went like, hey, look, organisms produce many offspring, and they have great reproductive potential. But they reproduce more than necessary to maintain a population, and they reproduce more than can be supported by food supply or space, aka they don't really have family planning when it comes to animals here. But population size is surprisingly constant over time. That's interesting. So what is it, right? What is it that causes this? And the answer is, This is the first deduction. This is the struggle for existence. Individuals or members of same species must compete for survival. This is called intraspecific competition. So between members of the same species. Many die due to environmental factors and therefore do not reproduce because of the limited resources here. Okay, and selection pressure occurs. There's something in the environment that causes a difference in survival between individuals with different traits. So some of them survive and some of them don't. And this is why, like butterflies here, they can produce many offspring, but only some survive, and the population number stays around the same number. Okay, the population stays around the same size. So this is the first deduction. What else did they observe? So they also observed that, hey, there is variation in the phenotype. And this is caused by genetic variation, primarily mutations, as we talked about last videos, which introduces new alleles. Now, combining our previous observation and our deduction and this observation, we can then conclude that, hey, there is the survival of the fittest. Some individuals, all individuals look somewhat different, right? Some individuals have advantageous alleles, which are better adapted to survive, or we say that they have a selective advantage. Okay, I want you to pay attention to the English words used here because it's a different language, but to describe what's going on here. So some individuals have advantageous alleles, they're better adapted, and these individuals would have then higher fitness. What do we mean by fitness? We don't mean that these butterflies here, which has variation, some of them go to the gym more than others. No. We're saying that they have higher fitness refers to they are more likely to survive and reproduce. Higher fitness means some of the butterflies with these advantageous alleles are more likely to survive and have more offspring. And as a result, has a higher chance of passing on these advantageous alleles to the next generation. We say that these advantageous alleles are selected for, whereas the disadvantageous alleles, the others, are selected against. Okay, I want you to pay attention again to the language, especially this word more likely. Now, why more likely? Because it's not definite that they survive. It's not like, oh, you're a red butterfly, you 100% survive. You're just more likely. Okay, so an example here, maybe the predator cannot see red as well, but can detect blue very well. And as a result, more blue butterflies die. Okay, but it's not a guarantee that if you're blue, you will die. It just makes it more likely that you will die. Okay, it doesn't mean if you're red, you wouldn't die, right? It's just less likely. Okay, so you can see here that even though some red and some blue died, more blue died as a result. They are more likely to die than the red ones, and the red ones, okay, we can say that they have a higher fitness as they are more likely to survive and reproduce more, and have more offspring, and therefore pass the red alleles to the next generation. So over time, over many generations, you can expect this. Changes in allele frequency, because the red pigment in the butterflies, there's an allele coding for it, right? So the red color allele is advantageous as being passed on, is more likely to be passed on to the next generation, and therefore the frequency of this red allele would increase over time. Now this results in some sort of adaptation, this red color, because it wants to evade the predator, right? A lot of these butterflies would have the red color, which is a result of natural selection over time. And this would enable it to be best suited to the environment it's living in, to survive with the predator around. Now over time, over many generations, okay, if this particular population in this specific place, okay, changes so much that it cannot it's completely different now from other butterfly populations, that can lead to a formation of a new species. If this particular population here is so different from the original one. Okay, but more on this later. Right, so the idea again is this, right, in conclusion. I'm going to go back to my title slides. Where's my title slide? There you go. So, different examples, same idea. Number one, there is some sort of variation, and some traits are beneficial and some traits are not. So in this case, the black mice are more camouflaged than the yellow mice. Okay, and the black mice are better adapted to the surroundings than the yellow mice. And therefore, the black mice are more likely to survive and reproduce and have more offspring, and therefore the black color allele that codes for the mice fur will be passed on to the next generation. It's very important to say not black fur is passed on, but black allele is passed on. It's a gene that is passed on, not the fur directly. And therefore, after the next generation, because the black mice produce more offspring, this will reduce in more black mice in the offspring. Okay, and therefore over time, the frequency of the black color allele has increased in this particular population, in the particular gene pool of this population. And we will do a few more examples later. But first of all, let's talk about the importance of variation. Variation, again, means the presence of different characteristics. We learned this in the previous lesson, right? Variation is present, okay? It means different individuals might be different, resulting in different survival rates when there is an environmental factor. So some survive and some don't, and some successfully reproduce and some don't. Okay, but this is very important because when there is a changing environment, at least some of the individuals in the population may be able to adapt to it. If everyone's the same, everyone would have the same tolerance or resilience towards a changing environment. But when there is variation, some individuals may survive and reproduce and enables that population. So again, zooming out, not looking at the individual, zooming out, that population can then survive because of that few different fellas, despite the changing environment. Now, think of it the other way around again. If there is low genetic diversity or low genetic variation, they might be more susceptible to disease, as when the disease appears, which no one has an immunity to, right, it could wipe out the entire population. So more on this next chapter. I just want you to know, again, that variation is very important for the population to survive despite a changing environment. Okay, so that's natural selection and importance of variation. There are actually different types. So when there's selection pressure, okay, selection pressure is an environmental factor that again causes some to survive and some to die. Okay, when there's selection pressure, something exerting pressure, natural selection occurs. That favorable allele, the advantageous allele, will always be selected for, and the less favorable allele will always be selected against. The one that is selected for would have an increase in allele frequency, whereas the less favorable allele will have a lesser allele frequency, a lower allele frequency in generations to come. Now, there are three types of natural selection. So, three ways the natural selection can affect allele frequency. So, number one, stabilizing selection. Number two, directional selection, and destructive selection. You can see in the dotted lines here, this is the original population. Dotted lines, original population. We assume that it's a bell curve. But when natural selection acts upon it, okay, different phenotypes are selected for and different phenotypes are selected against, resulting in different distributions. So let's look at stabilizing selection first. You can see here the intermediate phenotypes are selected for. So somewhere in the middle of the graph, this will increase in frequency. So this is increase in frequency, whereas the phenotypes extremes are selected again, so they are lesser here. In directional selection, only one extreme characteristic is selected for. So the curve sort of shifts to the right here. Okay, why? Because this extreme is selected against, and in intermediate is also selected against, whereas the more extreme phenotype is selected for. Next, in disruptive selection, it's the opposite of stabilizing selection, really. Both the extreme traits are selected for, whereas the intermediate traits, you can see here from the original curve, the allele frequency has definitely decreased, as the intermediate traits are selected against. So quite cute, right, the graphs? I like the graphs a lot. Anyways, let's move on to the next part, and let's think of a few examples of what's going on. So let's talk about stabilizing selection first. So a curve that goes from like this to like this. Ooh. Well, I wrote that wrong. Okay, so it's a higher here and lower on the sides. So let's give you an example. Let's talk about flowers. If the flower is too short, right, the flower plants, it might die because it may not be exposed to sunlight. But if the plants are too tall, it may be blown away by the wind, so it might die quickly. Where the medium flowers here may have the perfect balance of sunlight and resistance to wind damage. Let's talk about babies, weight of babies, mass of babies. If the baby is very light, it tends to have a higher mortality rate because lightweight babies usually mean that the baby is weaker. Not necessarily, but more likely, okay, like probable. Average weight babies survive more often and therefore are more healthy. But heavyweight babies, so not that they lift weights, right? They are heavy. They may have a higher mortality rate when being born, as it's very hard to push the baby out and they might die during labor. So in this case, right, you can see how natural selection influences the allele frequency. It selects against the extreme phenotypes and selects for the intermediate phenotype, resulting in the shape of curve, this to this. Okay, number 2, directional selection. So the curve moves to one side. Okay, it could be either way. They are both directional. If it shifts right or shifts left, it's directional. Okay, so here's this directional selection. This is actually a really bad example because it might not be true, but it's a commonly used example anyway. So a long time ago, there were giraffes with short and long necks, so there's variation. But the tall giraffes can reach the leaves, whereas the short ones and even the medium height giraffes, they cannot reach. And therefore, only long-necked giraffes survive, and therefore, over time, the long-necked giraffes have longer and longer necks. So this is directional selection. Short-necked giraffes are selected against. Middle-necked giraffes are also selected against. The environment, okay, natural selection favors the long-necked giraffes. Number 3, disruptive selection. So you might be thinking, now explain this. So how does it go from this to this? McDonald's. So sometimes in environment, right? In this case, there are 2 birds here with different beaks. Okay, and they occupy different niches. Okay, or how do we put it? They actually eat different food to adapt to their surroundings. Okay, so the big ones would actually look for these large seeds and can open those tough shells. The small birds with the small beaks can be able to handle small seeds and crack them open and eat them. Whereas, can you imagine if you have a medium-sized beak, you won't be able to open those tough shells and you cannot handle small seeds very well. It's quite clumsy. And therefore, the medium-sized beak bird may decrease in frequency. therefore resulting in a distribution that looks like this. Okay, so the extremes, big beaks and small beaks, are selected for, whereas the intermediate beaks, the medium-sized beaks, are selected against. Therefore, a decrease here in the intermediate phenotype. So I hope that gives you a better idea of types of natural selection. Okay, moving on to some examples. So I used the butterfly example, we talked about the mice example, so those are like quite sort of just very simple things. But here are examples which are real life. Okay, we learned 3 here, you need to know this very well. Number 1, antibiotic resistance, which you have learned about a little bit. Number 2, industrial melanism of a peppered moth, which is a very classic natural selection sort of example. And number 3 is sickle cell anemia. Now, more importantly, as we go through these examples, other than the details, you also need to learn the language of how to describe natural selection, because in the exam, what they would do is they would give you a scenario and they might ask you to explain it in terms of natural selection. So here we go. Let's talk about antibiotic resistance in bacteria. Now, good news is you've already learned this before. In chapter 10, remember? Okay, we learned that antibiotic resistance is when antibiotics are no longer effective against bacteria. For example, there's penicillin that inhibits cross-link formation between peptidoglycans in the cell wall. But some bacteria have penicillinase enzymes that can break down penicillin and therefore become resistant to penicillin. So what is it caused by? Number 1 is caused by spontaneous or random mutation in some bacteria. Okay, this is random. Very random, and this results in a new allele that may cause change in protein or production of new protein that cannot be targeted by the antibiotics. Now, maybe it starts with 1 or 2 bacteria, but natural selection enables these resistance genes to spread, allele frequency to change. How? Okay, so number 1 is random mutation. But number 2, when the person consumes antibiotic acts as a selection pressure. Bacteria of this allele coding for penicillinase, so bacteria that produces the penicillinase enzyme, has a selective advantage in this case, and therefore they are selected for. Antibiotics only kill bacteria that are non-resistant, whereas the bacteria with penicillinase are more likely to survive. Okay, you can see here that not all the bacteria will die as well, maybe just by chance. Okay, not 100%, but most will die. Okay, anyways, resistant bacteria are more likely to survive and reproduce, okay, over many generations, and pass down the allele to the offspring via binary fission. This is bacteria, right? So they have more offspring. They pass down the allele, and therefore you result with a lot of bacteria with that resistance allele. So over time, those resistant allele frequency increases, and now you end up with a population of bacteria which are all resistant to the antibiotic that you are using. You can see the trend here now. It's always talking about some sort of variation, mutation. It talks about the selection pressure next. Number 3, it talks about whether it survives or reproduces. And number 4, talk about allele frequency. It's always these 4 main bits. Let's do one more example. Industrial melanism. So this sounds very fancy, but actually we're talking about peppered moths. Peppered moth, or Biston betularia, no big deal, just scientific name thrown at you. They usually spend the day resting on tree barks or branches or trunks. And you can see here that they actually camouflage quite well. This is on a white tree, more white. This is on a black surface. And this is actually a camouflage to protect itself against its predator, which is birds. Okay, this is called a speckled appearance, and this is called the melanic or black appearance. Okay, this speckled. This one is melanic or black. Okay, but alright. So with these two fellas, in 1849, moths with a speckled appearance was more common. But, you know, number of moths with a melanic or black appearance actually increased in industrial areas during the Industrial Revolution. Other places where industrial areas are smaller, or then don't have industrial areas at all, the speckled form of this moth is still more common. Why is that? Because natural selection has occurred. Okay, how? Let's talk about it more. Number one, let's talk about variation. There's variation that's present, maybe due to a mutation long time ago. There are black moths and there are speckled moths. The selection pressure here is predation by birds. And the interesting thing is it happens more in industrial cities because the tree barks have a darker shade due to a high amount of pollutants and soot. At the time, the factories didn't have a lot of environmental policies. Okay, so the trees were covered in black soot and not a lot of fungi. This is actually caused by lichen. This appearance, oh no, that is a moth. It's caused by lichen, a type of a mixture of fungi and organisms which grow on the tree bark. Right, that's all gone now in the industrial cities. And therefore, you can see that this white moth now stands out, whereas the black moth is well camouflaged. And therefore, black moths, in this case, have a selective advantage as they were more camouflaged, and white moths are selected against. Selection pressure. Next point, you guessed it, is about reproduction. Okay, so black moths are more likely to survive and reproduce and pass on the allele to the offspring. And therefore, over time, the allele for black color increased in frequency in industrial cities, and there were more black moths in industrial cities instead of a speckled one. Ta-da! And that is an example of industrial melanism in peppered moths. So one more particular example before we end. So sickle cell anemia and malaria, actually. So frequency of sickle cell anemia is highest in areas where malaria is common. You can see here the green regions here is places that are affected by malaria, and these other really intensely red regions is where the sickle cell allele frequency is most common. You can see they are common in these areas. They correlate with each other. Now, why is that? So let's look at it. So actually, the sickle cell anemia, right, and malaria both acts as selection pressure. So how so? So let's look at the different genotypes here. There are three genotypes. Number one is homozygous for sickle cell alleles, HS. Now you might ask me, means what didn't you say we must use HbA? Now both are accepted in the exam, I've discovered. Okay. Anyways, if you're homozygous for sickle cell alleles, the red blood cells cannot carry oxygen very well, and the person may die from sickle cell anemia. So it's selected against. Okay, it's sickle cell anemia selects against is homozygous for sickle alleles. You also could be homozygous for the normal hemoglobin allele, resulting in perfectly normal red blood cells, but as a result, Plasmodium parasite can affect the red blood cell, and malaria is lethal. It can be lethal. So the person may be more likely to die from malaria if they are homozygous for the normal hemoglobin allele. So malaria selects against this particular genotype. What if though you're heterozygous for the sickle cell allele, so HNHS? Now this means you have the sickle cell trait and that you are a carrier. You do not die from sickle cell anemia because the person won't have normal hemoglobin as well as some sickle cell hemoglobin. The person will be less likely to suffer from severe effects of malaria as well, because Plasmodium cannot survive like this. Okay, it contains less Plasmodium in their blood if they get infected. So people who are heterozygous for sickle cell allele often have a selective advantage and is more likely to survive and reproduce and have more offspring, and therefore more likely to pass on both H and HS. As a result of that, sickle cell allele is maintained within that population. You realize here I didn't say increase but maintain, because you would assume that sickle cell allele is a bad thing, isn't it? It should be decrease. But when there's malaria around, it's maintained within population, because again, sickle cell trait individuals do not die from SCA, and do not die from malaria. And that's it for this particular examples. So just one more thing before we end. The Hardy-Weinberg Principle. So this actually combines your knowledge from genetics, last chapter, and the natural selection of this chapter, and use this knowledge to actually calculate allele, genotype, and phenotype frequencies in these populations. Now, this is a theoretical equation sort of thing, so I'll show you the equations first. It looks like this. We'll talk about it later, but you can use these equations here actually to actually estimate how many alleles, okay, or what the allele frequency is in the population. But there are 5 requirements because it's theoretical, and this principle assumes that there is a large population so that the ratios are about right. Number 2, there's random mating, there's no biasness in that. Number 3, there is no emigration, so no one moving in or out, no mutations, no selection pressure against any one genotype. So if we take our theoretical values that we calculate from the particular equations, and we look at the environment, and if it matches, this means 5 of these requirements are met. But if we calculate these theoretical values and compare it with the environment, and we realize that it doesn't match, this means that one of these requirements, at least one of them, are not met. And there may be some selection pressure somewhere, or some mutation somewhere that's going on, or maybe some moving around, or maybe the population is just too small. Okay, so that's the Hardy-Weinberg principle and requirements of the principle. Let's go to the equations, which is the fun part. So again, the Hardy-Weinberg equations or principle is actually used to calculate the frequency of alleles, the frequency of genotypes, and phenotypes, if you know what those genotypes code for. Okay, so number 1 here is actually representing all the alleles in gene pool. So the first equation actually makes sense, right? If you sum up all the dominant alleles, so frequency of dominant alleles, and the frequency of the recessive alleles here, if you sum them all up, you will result in all alleles in the gene pool. 1 is like 100%. But instead of 100%, we write 1. Okay, it's in that kind of format. Equation 2 is for genotypes. Okay, and it's actually derived from this little Punnett square here, crossing of 2 heterozygous individuals, forming this 3:1 ratio here. Okay, and we can see this in the equation. We can see that it makes sense here because homozygous dominant is big A, right? And dominant allele is represented by P. So P square, so 2 big A's together, that's P square, is representing the frequency of homozygous dominant individuals in the population. Let's look at heterozygous. It also makes sense. There are 2 fellas here in the Punnett square, right? So that's 2 there. Heterozygous is 1 dominant and 1 recessive allele together, so P cube, 2P cube. Q squares also makes sense here, as it's homozygous recessive. Okay, so 2 recessive alleles, so which is represented by Q, so Q square. And add all this together, you should get all the alleles in the gene pool. Ta-da! So let's put these equations to good use. So we're going to do 2 examples here in this video. I'm going to show you the example, and I hope that you are able to pause the video and try it on your own before you play the video again. Okay, try it out so that you know how to go about it later on. Given that the incidence of the genotype small a is 16%, calculate the frequency of dominant allele big A and the frequency of homozygous dominant individuals and heterozygous individuals. So what is small a here? Small a, okay, is actually Q square. Q square, right, as we saw just now in the previous slide, right, shows us, again, the individuals, how many individuals that are homozygous recessive in the population. So we convert it to 0.16. We want it in this format. And using that, we can find the following things, right? Dominant allele A, right? So big A here is actually represented in the equation P. And what is P? According to the equation P, okay, move Q to the back, so it's 1 minus Q. So that is 1 minus square root of 0.16, which equals to 1 minus 0.4, which equals to 0.6. So frequency of dominant allele A is 0.6. Now how about the frequency of genotype big A, big A? Now this is represented by P square in the equation. Okay, remember P square is homozygous dominant. And from the previous question, we found it already, so it's 0.6 square. There's 0.36. Last but not least, we can find the genotype big A, small a frequency. Yeah, this is the frequency of heterozygous individuals, and that's represented by 2pq. So how to calculate 2? Right, we already found P just now, 0.6. And we already found Q just now, this is Q, square root of 0.16, which is 0.4. And you get 0.48. Ta-da! And that's how you calculate, that's how you use the equations really. Let's do another question. This is another example. And again, I would like you to pause the video after you see the question and try it on your own before you look at discussion. Okay, this is from this is a past year. This is about lactose intolerance, industrial persistence, which is the opposite of intolerance, and a mutation that causes it is here. So people with lactose intolerance have genotype small t, which is homozygous recessive. Lactose persistence have genotypes big T, and big T, small t. They tested 166 people, and 58 people were found to have lactose intolerant. So calculate the frequency of allele big T. Sure, working. So how do you do this? So small t here is actually represented by Q square, right? This here, right, is P square, and this here is 2pq. They're telling us that 58 out of 166 have lactose intolerance. This is small t, which is Q square. Okay, so what is Q square? Q square is the frequency. Is it 58? No, it's too big. It has to be below 1. They tested 166, so 58 out of 166 lor. Okay, and with this you can find out frequency. Let me type it in my calculator. Right, that is 0.3494, somewhere there. Okay, so from here we can then calculate the frequency of allele T. Allele T, big T is P. It's a dominant allele here. So how do we find that? So let's find Q first, which is the square root of 0.3494, which I will need to type in my calculator right now. Square root of 0.3494. Okay, there you go. But you can't see that. Anyway, that is equivalent to 0.5911. Okay, the final answer should be in 2 decimal points, but I'm giving as much decimal points here to make sure it's accurate enough. So how do you find P? So P here is 1 minus Q, so is 1 minus 0.5911. 1 minus 0.5911 is 0.4089, which is, okay, 2 decimal points, 0.41. So ta-da! This is the frequency of allele T, big T, dominant allele. And that's it. So today we've pretty much done all these bits here. We have done what is natural selection, the genetic variation in selection, importance of that, the 3 types of selection, all the different examples, and the Hardy-Weinberg Principle. So we are done with this part, which is all part 2A. The following will be part 2B. That's it from me for now. See you next video.